Hello everyone, myself Professor Shahana Sengupta. Presently, I am associated with Swami Vivekananda Institute of Modern Science, Kolkata as an assistant professor in the Department of Microbiology. I would like to welcome you all in my YouTube channel. In my YouTube channel, usually I share different types of academic videos related to the field of biological sciences. Today, I have picked up a topic for you all, that is biofertilizer. It is a very burning topic in today's scenario. Let's start our journey with a new topic. Happy listening. Let's begin with Vesicular Arbuscular Mycorrhiza or VAM and its application as a potential biofertilizer. The term mycorrhiza was taken from Greek language meaning fungus root, and the term was coined by Frank in 1885. The mycorrhiza is a mutualistic association between fungal mycelia and plant roots. VAM is an endotrophic fungi. That means it lives inside the body of the host and generally formed by acceptated Phycomycetes fungi. It colonizes the roots of approximately 80% of plants' body. VAM help in nutrient transfer, mainly of phosphorus, zinc and sulfur. They also mobilize different nutrients like copper, potassium, aluminium, manganese, iron, magnesium from the soil to the plant's root. Let's focus on the morphology of VAM fungi. They are composed of three basic units: external hyphae, arbuscles and vesicles. The hyphae is known as extra-radical or external hyphae that are filamentous in nature, which ramify through the soil. They are responsible for nutrient acquisition and propagation of the association as well as spore formation. VAM fungi produce different types of soil hyphae, including thick runner or distributive hyphae as well as thin absorptive hyphae. The finer hyphae can produce branched absorptive structure where fine hyphae proliferate. The network of hyphae in the soil is only connected to roots by the entry points that initiate mycorrhizal association, as hyphae do not grow out of the living roots. Arbuscles are intricately branched structure that formed within a root cortex cell. They look like little trees. Arbuscles are formed by repetitive dichotomous branching and reduction in hyphal width, starting from initial trunk hypha and ending in a proliferation of fine branched hyphae. The vesicles are sac-like structure for storage of nutrients. They can develop thick walls in ordered roots and may function as propagules. Some fungi produce vesicles which are similar in structure to spores. They produce in soil, but in other cases they are different in nature. Let's discuss about the mechanism of action of VAM. The VAM forms an association with plant roots. Mycorrhizal fungi basically establish a mild form of parasitism that is mutualistic. That means both the plant and the fungus usually get benefit from the association. About 90% of land plants rely on mycorrhizal fungi, especially for mineral and nutrient transfer. In turn, the fungi receives nutrients from the plant. It penetrates in the root cortex and spreads around the root of the plants. As the name indicates, the sac-like vesicular structures of VAM store phosphorus and phospholipid nutrients. The other structure, called arbuscles, help bringing the distant nutrients to the vesicles and the root region. Let's focus on the action of mycorrhizae. It enhances the feeding areas of the plant root as it is spread around the roots. It also mobilizes the nutrients from distant part to the roots. It can store nutrients. It also can remove the toxic chemicals like phenolics from the soil, which otherwise can hinder in nutrients availability as well as nutrients mobilization. The VAM hyphae is also helpful in retaining moisture around the root zone of the plant. It also can increase the resistance to root-borne or soil-borne pathogens and nematodes. Now, I would like to discuss about the mass production procedure for VAM. For the mass production, first of all, the spores of VAM are isolated. After that, the spores are mixed with sterilized soil. Next, the sterilized soil mixed with spore is introduced in a sterilized pot, followed by the host plants are introduced in the same pot and kept in that situation for 3 to 4 months. The whole system can be kept in greenhouse for further progress. In the very next step, the soil in the pot along with roots of the host plant is macerated. The step is basically performed to increase the organic quantity or organic load in the soil. After that, the soil is dried till it attains 5% of the moisture content. After that, the dried soil inoculants are used for the field application. That time it can be used as a very potential biofertilizer. Let's talk about phosphate solubilizing biofertilizer. Phosphorus is one of the most important plant nutrient and may be critical nutrient for the optimum growth of plants. There are many types of microorganisms present in the soil, out of which some microorganisms and fungi can solubilize the complex insoluble form of phosphorus into simple soluble forms which can be taken up by the plants. In the rhizosphere of crop will render insoluble soil phosphate available to plants due to production and secretion of organic acid by themselves. The use of these biofertilizer will also increase the availability of phosphate from rock phosphate applied directly even to neutral to alkaline soil or when used for preparation of phosphor compost. Generally, these microorganisms are very few in soil. These soil phosphate solubilizing bacteria stay near the roots and make up phosphorus available to plants from soil as well as from fertilizer and increase the production drastically. Phosphate solubilizing microorganisms include efficient strain of bacteria, fungi, yeast and actinomycetes. Let's focus on the ways of application of biofertilizer in the agricultural field. There are 3 different ways of application of this biofertilizer in the agricultural field, namely seed treatment, root dipping and soil application. Let's begin with the seed treatment. Seed treatment is the most common method adopted for all types of inoculant. The seed treatment is effective and economic too. Seed treatment can be done with Rhizobium, Azotobacter, Azospirillum along with phosphate solubilizing microorganism. It is having no side effect at all. Firstly, the seed must be coated with Rhizobium, Azotobacter or Azospirillum. When each seeds get a layer of above bacteria, then the phosphate solubilizing microorganisms inoculant has to be treated on outer layer of the seeds. This method will provide maximum number of population of each bacteria required for better result. Mixing of any two of the bacteria and the treatment of seeds will not provide maximum number of bacteria of individuals. The second important part is root dipping. Application of Azospirillum with the paddy and vegetable plants can be done under the method is needed. The required quantity of Azospirillum has to be mixed with 5 to 10 liters of water at the corner of the field, and all plants have to be kept for minimum half an hour before sowing. The third process is soil application. Phosphate solubilizing microorganisms has to be used under this process. The proper ratio is use of 2 kg of phosphate soluble microorganisms per acre. Phosphate solubilizing microorganisms is mixed with 400 to 600 kg of cow dung along with requisite volume of rock phosphate if available. The mixture of phosphate solubilizing microorganism, cow dung and rock phosphate have to be kept under any tree shade or shielding for overnight and need to maintain at least 50% of the moisture content. Then the mixture is used in soil and in rows or during the leveling of the soil. Production and utilization of good quality carrier material is another aspect of biofertilizer. The use of ideal carrier production of good quality of biofertilizer is very important in this regard. Peat soil, lignite, vermiculture, charcoal, press mud, farmyard manure and soil mixture can be used for the same purpose. Neutralized peat soil or lignite are found to be the best carrier materials. Ideal carrier material should have some characteristics like they must be cheaper in cost, they should be locally available, higher organic matter content should be there, they should not be toxic, water holding capacity will be there more than 50% and they must be easy to process. For the preparation of carrier material, neutralized and sterilized carrier material is spread in a clean, dry, sterile or metallic sheet or plastic. After that, bacterial culture drawn from the fermenter and is added to the sterilized carrier and mixed well by manually or mechanically. Inoculants are packed in polythene bags and sealed with electric sealer. After that, it is ready for the market. Let's discuss about some beneficial aspects of biofertilizer. Biofertilizer is the key element of sustainable agriculture and organic farming. Microbes in biofertilizer provide atmospheric nitrogen directly to plants. They aid in solubilization and mineralization of other plant nutrients like phosphates. Beta synthesis and availability of hormones, vitamins, auxins and other plant growth promoting substances can improve plant growth. It also permits increased crop productivity, promote plant growth by incorporation of essential nutrients. It also can facilitate implementation in different types of soil. It can reduce runoff and obtain economic benefit for farmers. It is eco-friendly in nature. Biofertilizers also can improve the soil's texture, allowing it to hold water for longer period of time. And it can also increase the bacterial and fungal activity in the soil. So they not only assist plant but also help to improve the soil quality and crop production. It can be a good substitute to the chemical fertilizers also. Thanks a lot for your patience. Please do subscribe my channel. Stay tuned for the upcoming videos in different fields of biosciences.